So we're going to do a quick recap over mitosis, which you guys learned last year. This is where the cell makes an identical copy of itself. So we see this in cloning, we see this in reproduction in some animals, and for us, chiefly we see it in our ability to heal, grow, and develop. So again, mitosis is what is called asexual reproduction, where two identical copies of a cell are made, and this does not need the cell of another organism to mix with in order to reproduce, like we do. So we have two identical cells in the end, and basically it's a cell making an identical clone of itself. And so what we're going to do is remember what a eukaryotic organism or eukaryotic cell is real quick. Eukaryotic cell has the nucleus in the center. So the chief thing to know about how a eukaryotic cell makes a copy of itself is it starts from the inside and goes out. So first the nucleus, everything inside of it has to copy itself. Then the nucleus makes a copy of itself, and then the cell can make a copy. So here's how that is shown on a little bit of a diagram. So here we have our first nucleus. Each the nucleus has 4 chromosomes in it, and then they replicate here. So now we have still 4 chromosomes, but each one has a copy. And then the nucleus is actually going to split into 2. So basically an intermediate step here would be 2 nuclei, and then the cell would start to split here into 2 cells. So here we get a little bit more specific. Here's our 2 new nuclei, but the cell has not yet completely split. So it's very close to doing so, but it hasn't made it all the way. The thing to route out here is that a lot of people think that what's called the cytoplasm is this fluid here. That's not entirely true. The cytoplasm is a region of the cell that is everything outside of the nucleus. So anything within the nucleus is called the nucleoplasm. So that's the inner region. The cytoplasm is the outer region, including the cell membrane or wall, depending on if it's an animal or a plant cell, and the cytosol, which is the fluid that surrounds the nucleus. And cytokinesis, hence, would be the splitting or the movement of that cytoplasm apart into 2 new cells. So mitosis has 5 phases: interphase, prophase, metaphase, anaphase, and telophase. And the new process of meiosis, which is how cells are sexually reproduced, has the same phases, so you don't really need to learn any new terms. But we have a quick acronym to help us memorize those phases. Idiot, pass me another taco. So if we look at the first letters here, I, P, M, A, and T, those are the same first letters and order of the phases of mitosis and meiosis. So remember, idiot, pass me another taco. Tasty. So we go back to the I, which would be interphase. Interphase is where the chromosomes make a copy of each other. So first, all the DNA in here has to duplicate. Again, we start from the inside out before the cell can split. And here's actually what's happening. So in our little equipment here, we have our pipe cleaners, which represent chromosomes. And each one has a little white bead on it. So I'm going to color that in real quick. White bead. Now your white bead represents what is called the centromere. And what happens is that your chromosomes actually make a copy, and they duplicate to where we have 2 arms coming out of it. So it's kind of like a stick figure without a head. And the new term that we need to understand here is that when the chromosome replicates itself to look like an X with a little white dot in the middle, which is called the centromere again, each arm or each side of it, okay, that's a side. Let me highlight it here. That's a side, and that's a side. It's called a sister chromatid. So that's a sister chromatid, and that's a sister chromatid. So when you see sister chromatids, that's replicated chromosomes. Okay, interphase is our first phase, so that's where the DNA is replicated, and the DNA basically makes a large, uncondensed mess of itself as it duplicates, and this is called chromatin. And then it'll be organized more neatly into sister chromatids. Prophase, so we're on the pass of my idiot pass my taco. Pass me a taco, excuse me. The chromosomes are actually duplicated, and we have the more organized version. So here we have our sister chromatids in prophase, and we have our biggest chromosome of dad, who is red, and our biggest of mom, who's blue. So they get close together. Our next set from mom and dad, and our third set from mom and dad get close together. They're just kind of hanging out in the cell. Then in metaphase, they get more organized. So the biggest line up at the top in single file, the next biggest line down the middle, and the smallest line down the middle. And basically, we have these centrioles over here that they're like these fishermen. So basically, they take these fishing poles, hook onto each sister chromatid, and they're going to break them apart so we can split the cell into two. So this is our nucleus beginning to divide. So then the centrioles in anaphase take our sister chromatids, fish them apart, and now they bring them towards each pole of the cell. So now we see the cell start to stretch in the middle. So this is where our nucleus begins to divide. And then now we have two nuclei. So our nucleus has divided in telophase. So now we're in the taco of idiot pass me a taco. And the nuclei have divided. So now that the DNA has replicated, organized, and separated, now we can start to break the cell. So the innermost step of this is complete. Now the outside can now split. So if we look here, we have an exact replica in this nucleus of the original cell that we started out with. We have 6 chromosomes, 3 blue of different sizes and 3 red of different sizes. And so in the end, we will have 2 identical copies of our original cell. And remember, this is our example of asexual reproduction. So bacteria reproduce this way, which is why they evolve so quickly. Yeast does this, so we used yeast when we made our root beer and our sodas in chemistry. And plants also can asexually reproduce. And here are 5 different types of how this happens. So budding is when cells just kind of make another. Binary fission. The parent divides equally in results, which is what we just did. So our parent divides equally instead of one smaller cell just being born from the parent. Spore formation. So this is how fungi reproduce. Cells will just germinate when released from the parent. So they're usually covered by like a protective coating for preservation. So we also see this in plants, and such as vascular plants and ferns. Regeneration and fragmentation. So we saw this in the newt and the axolotl and the starfish when the organisms were able to like lose an arm and regrow a new organelle or organ, such as the heart or a portion of the brain. And we can also do this to a certain extent when we heal and grow new body parts. And then vegetative reproduction. So again, that's going back to plants and roots and stems and leaves. So our punchline is, remember that mitosis is just cell cloning itself. Remember its chief purpose. Remember those steps in that acronym, idiot pass me a taco. IPMAT, interphase, prophase, metaphase, anaphase, telophase. And remember how to diagram each step.